Welcome everyone. Thanks everyone for joining for the second installment of a 6-week summer faculty lecture series. These 1-hour virtual lectures are scheduled every 3 weeks to showcase wonderful Williams professors from a variety of academic fields. The first lecture with Professor Dara Goldstein focused on Russian culture and cuisine. You can find listings for any upcoming Williams events and recordings of past talks if you visit alumni.williams.edu/virtual. The Williams Chicago Regional Association is delighted to act as the local host for today's event. My name is Janae Jackson. I am class of 2010. I am the president of the association and I've volunteered in this position since 2015. We are excited to see so many Williams alumni and parents from across the country, in addition to a lot of East who joined from Chicago today to join us for this virtual gathering. One of the big highlights for the Williams community in Chicago is the annual lecture, every spring hosted by Peter Pond, class of 67. Obviously, we're not able to safely gather in person for our traditional lecture. So we're excited to extend this event concept initiated by Peter and Alicia Pond to this virtual format for this year. I'd just like to express my thanks again to Peter for your generosity in hosting the annual faculty lecture in Chicago. It is always a very wonderful and sophisticated event, and we look forward to being able to join you guys again in 2021. As for this year, after today's talk, the next upcoming virtual Williams faculty lecture will be from political science professor Sam Crane on July 23rd, and then geologist Alice Bradley on August 13th. So keep those in mind. You can register for those as well. Keep your eye out for any announcement with additional information in the near future. Lastly, I want to take a quick second for some housekeeping and tech notes for everyone. The chat feature is on, the chat feature will be turned off during the Zoom webinar. We will not be able to chat during the Zoom webinar. However, I would like to encourage everyone to submit questions to Professor Thuronyi by writing in the Q&A feature. So you'll see a Q&A feature on your screen. You can tap that feature. You can type in your question anytime during the lecture. You do not need to wait until the end of the lecture to type in a question. Ben will be able to verbally answer questions at the end of the lecture, so no need to wait. He will field all the questions at the end, and feel free to submit anything during the talk as you think of them. With that, I would like to welcome Peter Pond, who will introduce the wonderful Professor Thuronyi.
Thank you, Janae. It's a pleasure to be here. As Janae said, my wife and I like to display what's best about Williams College by having lectures by prominent Williams professors. Today we have Ben Thuronyi, who is one of the young superstars in the chemistry department. Ben is a biochemist and synthetic biologist who joined the faculty of Williams in 2019. Ben studied chemistry and philosophy at Swarthmore College before receiving a PhD from UC Berkeley, researching how to incorporate fluorinated molecules into metabolic pathways with Professor Michelle Chang. Ben's postdoctoral work in Professor David Liu's group at the Broad Institute focused on directed evolution to make new genome editing tools, which is a very hot topic today, obviously. Ben's teaching interests at Williams span organic chemistry, biochemistry, and synthetic biology. The Thuronyi Research Group aims to develop an extremely fast-growing bacterium, Vibrio natrigens, as a synthetic biology workhorse biocatalyst. And with that, Ben, why don't you take over and talk about synthetic bio, engineering life. We're excited to hear your presentation.
All right, thank you so much, Peter, Janae, and thank you to all the organizers for the opportunity to speak to you today. I'm very excited to tell you all a bit about synthetic biology, a field that I love and have grown to know better over time. So I'm going to try to just touch very lightly on a whole bunch of topics today, not really do a deep dive into things, but I'm happy to follow up on any specific points in the Q&A, and I hope to just give you some food for thought. And really the whole talk is laid out on this slide, the philosophy of going from a living organism using engineering principles to decompose it and then put it back together into something new. So of course, I want to start this webinar in which none of us can see and hear each other with some audience participation, but bear with me. So here's a question for you. You can answer aloud if you want. You can just think your answer to yourself. Taking a look at this picture, what would you say is the most sophisticated thing that you can see? So probably not the digital SLR, maybe the phone, maybe the computer. You could have some controversy there. It's the plant. By a million miles, it's the plant. Can you imagine if your phone could heal its screen when you cracked it? And it could get all of its energy from the sun and from soil and water. And if it generated little phone seeds that made new phones for you, maybe next generation phones. So really the technology, if you're willing to call it that, and I certainly am, that exists in the natural world is kind of far beyond a lot of what we can do with sort of purely human innovations. And so synthetic biology is really all about harnessing and manipulating that technology, using it as technology to solve human problems. So I'm going to try in all of the parts of this talk to tell you about some of the challenges for that goal and some of the opportunities for it. I'm going to start by talking about GMOs, genetically modified organisms, because actually everything in the talk pretty much is a GMO. And I want to maybe push your thinking on those things a little bit. You can see how you feel about that. I want to talk about what I think synthetic biology is. There's some disagreement about that. And then touch very, very lightly on a bunch of things that people are using synthetic biology to do now and things that we might be able to do with it sometime in the future. Again, challenges, opportunities. And then I'll talk a bit at the end about how in my research lab, Williams College students are trying to contribute to all of those efforts. So first off, what is a GMO? Well, I would argue that humans have been making GMOs for a very, very long time. So we took things like the ancestor of modern corn, teosinte, and through selective breeding, we turned it into something that is practically unrecognizable in its modern form. We also took wolves and turned them into dogs. So we have both food and pets from selective breeding. And really selective breeding is just a very slow form of genetic engineering. These are heritable changes that we're introducing into an organism by choosing which of the offspring we choose to pass down their genes. So food and pets over tens or hundreds of thousands of years, what if you want faster results? So here on the left is a Hudson grapefruit. It's got a lot of seeds in it. It's got a certain color and taste. Maybe you don't think that's the best grapefruit that it could be. Well, one option is take some of those seeds and blast them with gamma radiation. So then you get the star ruby grapefruit. Probably many, many of you have eaten a grapefruit that was produced by irradiating these seeds and then just picking, oh, I don't know, what's gonna happen? Let's get random mutations. And this one has fewer seeds, it's different color. Let's try it and see if it's good to eat. And this kind of thing was so popular starting around the 1940s that there was, in fact, the Atomic Gardening Society, where people did this for fun and would get certificates for their irradiated plants. So this is a way to speed up those processes, but I would argue there may be some disadvantages to not knowing exactly what changes you're introducing, but this is how we've done things for a long time. So food, companionship, what about medicine? So many people need insulin in order to survive. It's a non-negotiable item for them. And we used to get that from literally processing tons of pig and cow organs. So I was horrified to see this, the caption for these photos. Pancreas glands are examined as they arrive from the meat packing house, then run through grinders. And this again is on the ton scale to get ounces of insulin out at the other end. And you can see some animal-sourced insulin down at the bottom. So maybe you want to do this in a somewhat more ergonomic way. You can take the gene that encodes human insulin, not pig or cow insulin, and put it into bacteria. And by inserting that gene into E. coli, sort of the lab workhorse bacterium, you can have those E. coli produce that protein. And that is actually the first genetically engineered medicine that was approved by the FDA. I'll note here that zooming in on the package saying human insulin recombinant DNA origin. So DNA has been taken apart and put back together in a new source. And actually since 2006, the only insulin you can get in the United States, and most of it worldwide, comes from the same kind of recombinant source. So I would argue that all of those things that I just talked about are GMOs in some sense, but they have different levels of sophistication, different scopes and scales. Putting a gene into a bacterium when that gene didn't used to be there, it's coming from a completely different kingdom of life, is a really different order of things from choosing which of your litter of puppies you want to have more puppies. So if all of those things are GMOs and you can think about which ones you think are and aren't, how are we going to feel about that? I feel pretty positive about sourcing insulin from bacteria instead of from pigs and cows. I think many of the people who protest GMOs might agree with me on that. There are other people who are very pro-GMO. So these are buttons. I have one in my office that were distributed by Ginkgo Bioworks, which is a synthetic biology company. And they're saying the kind of daring thing, which is, we want you to love GMOs. So really everything in the rest of the talk is a GMO of one kind or another. But I would say that the public conception of GMOs has really been strongly shaped by one kind, and that is GMO crops that have been engineered to resist pesticides. This is Monsanto, now bought by Bayer, Roundup Ready crops and their ilk. So these are things that let farmers plant crops and then blast their fields with pesticide to kill the weeds, but have the crops survive. But really, that's not necessarily any different from inserting a gene into a bacterium to make human insulin. It's on a continuum with selective breeding, random mutations and variations. Here's some tomatoes. This is another GMO that you might not be familiar with. This was the first GMO that was marketed to consumers in the US, the Flavor Saver tomato. These aren't actually flavor savers, but it illustrates the concept, which is normally a supermarket tomato has to be picked off the vine green. It doesn't have time to ripen because when you want to ship that to the supermarket, it needs to be very hard so that it doesn't bruise and break in transit. The Flavor Saver has one gene inactivated, and that gene, when you knock it out, it prevents the tomato from softening on the vine, so you can let it continue to ripen for however long you want and get better flavor. Actually, these things didn't work in the market, not so much because of GMO concerns, although maybe, but because they weren't profitable enough. But I, for one, would like to buy them. And then here's another example at the bottom. So these are two different salmon. One of them is a natural, unengineered salmon, and the other one has had a few genes from a different salmon variety and a gene from an eel species added to its genome. And that salmon now matures and is able to come to market size, within almost half the time that a native salmon does. So these things are all made in different mechanisms, and I would say it's worth thinking about what the social or economic effects of them are, other than just labeling them all with GMO. And again, where you put that label also is going to depend. So GMOs, synthetic biology is a little more than just GMOs. I would say it is a more sophisticated version of all of the stuff that I just showed you. Those are the most basic kinds of synthetic biology organisms that I just put up. Of course, what this field is kind of a matter of opinion. So, you know, early on in its conception, they asked 20 different synthetic biologists what they thought the definition of the field was. And you got 20 different answers, of course. So you're going to get my personal perspective on this, but I think it's somewhat mainstream. And of course, I should point out this field is only about 20 years old. The first paper you can really say is definitely a synbio project was only in the year 2000. So I would say that synthetic biology is a mindset. It's an attitude towards approaching living systems. And that attitude says living systems like bacteria, shown here, are not just something that we can study and understand and try to describe, but something that we can actually engineer. And that engineering mindset has a bunch of parts to it. So you look at a bacterium as an engineer and you say, this isn't just a bunch of goop in a bag on the micro scale, but this is in fact parts, circuits, chips, all kinds of things that you can understand design rules for and then reconfigure in ways that might solve a problem. So it's a very applied field, and it relies on something common to engineering, which is this design, build, test cycle. And some people also put in learn. I'm definitely in favor of that. So design, build, test, learn, and then design again. Because this is one of the huge challenges for synbio, is that despite the fact that we get to take advantage of all of this incredibly sophisticated, evolved technology from nature, we're not very good at engineering it. And in fact, it is systems that are not designed to be engineered, because they weren't designed in the first place, they happened. And there are properties that they have that make it very difficult to engineer them. So you often have to go around this design, build, test loop many times before you get to a solution to your problem. So I'll say synthetic biology hopes that living systems are engineerable, but I'm just aiming to make them that way if we can. So another way to look at that engineering mindset is to say, going from the very bottom level of DNA bases, that we now have a way of understanding the genomes of organisms. We can read out the DNA bases that encode all of the various behaviors that they have. We don't necessarily understand what all of those do, but for some of them, we can understand the function well enough to add a layer of abstraction. So rather than just going for DNA bases, you can now move up to considering those bases to be parts, things that have a defined function, they have modular behavior, and you can set them up into devices. So things that have an input and an output, and however they do what they do, you can understand what that is and then put them together into even bigger things, systems. So that all relies on principles like standardization. So that's everybody using the same kinds of units and ideas and communicating in a standard way. Abstraction, which lets you take away the molecular level detail and therefore start to build up much more sophisticated devices. And it relies heavily on the fact that we can put together DNA as well as read it. So I want to give you just a slightly more granular idea of what that might look like. Again, there's a lot of concepts on this slide, but really all you need is high school biology. So to get to the idea that a gene is a sequence of nucleic acids in the genome of an organism, let's say. And what I want to assert to you is that at least for fairly simple systems like bacteria, and somewhat for more complicated systems, but I prefer to stick to bacteria, we understand the ways that the elements of that DNA work well enough to actually label them, sort of decompose them into modular parts. And so within this stretch of DNA, which I'm showing you sort of schematically here, we can label sections of it as, here's a region that codes for a protein, this part gets transcribed and then translated, we get a biomolecule out of it. Here are regulatory elements that change the transcription, change the translation, change how the DNA is copied. It doesn't matter what the details are, but I'm trying to argue that if you understand those kind of design principles, you can take these things apart, put them back together, create a design, and have some idea that it's going to work out the way that you want it to. And here's just an example literally pulled from the computer-aided design program that my lab uses. Here is a zoom in on a promoter sequence. This thing initiates transcription of a gene, and it's just literally DNA bases. Here's the top strand on the top, and then you have a reverse complement strand on the bottom to make your double helix. And the real power of synbio, as advanced or unadvanced as it is that we have these labels. We can say, here are the bases, TTACG, and that corresponds to a certain functional element. If we really have good models and understanding of how these things work, we can take them apart, put them back together, do a new thing. And that really relies on all kinds of tools from synthetic chemistry, from biotechnology, things like PCR, the polymerase chain reaction that we just borrowed from nature, and engineering advances. And those things have meant that, especially in the last 10, 15 years, synbio has really relied on this, that we can make any DNA sequence we want. I can literally go to a company, I can type in, say, 50 bases in a row of whatever sequence, I can send that off, and within a day, if I pay for overnight shipping, which I don't usually, I can get that DNA in a tube, and I can use that to construct a design. So here's just a schematic of that. We make constructs of DNA bases on the computer. We buy synthetic chunks of DNA that let us put those things together using enzymes and other tools from nature. We put those into bacteria, and then eventually we can do our tests and experiments. And so what that amounts to is being able to take apart and reassemble DNA. And that's really supported by things I'm not going to talk about too much, but high throughput sequencing, so we can read entire genomes, fast DNA synthesis, and also CRISPR, which lets us put DNA into the genome of an organism directly. So that's just a very high level look at what synbio is, at least in my opinion. Now I want to tell you a bunch of very, very short stories about things people are doing or are trying to do or are working on now, or maybe are aiming to do sometime in the future. And hopefully give you more of a concrete idea of what the point of doing this engineering might be. So what can we do with synthetic biology? Lots and lots of things, potentially. I'm just going to touch briefly on 4 of them, and then there's a whole list of other things that I'm not going to talk about at all, though again, I'm happy to take questions. And if you want to look at this list a little bit, I just want to remind you, you can put questions into the Q&A chat. I've already got a couple anytime, and I will address those at the end, at least a few of them. So first thing I want to talk about is metabolic engineering. This is the idea that you can take usually a microbe and turn it into a molecular factory. So use its metabolism to generate some molecule that you want. And actually, bacteria and microbes are astonishingly good at doing chemistry. Usually we rely on the natural world in some way for a lot of the sophisticated molecules that we make. So the example I want to take is charged and interesting in all kinds of ways, and that is making opioids using yeast, actually literally baker's yeast. So the way that we get opioids now, and whatever you think about the opioid crisis and overprescription and so forth, these are a really valuable tool in the arsenal of modern pain relief. We get them from opium poppies, basically. All of the opioids that we use in the clinic are derived from literally collecting the sap of an opium poppy and boiling it down and extracting out the molecules. And then we do a little bit of chemistry on them. But by far, the vast majority of the chemistry, especially the really hard chemistry, is done by the poppy itself. Now, you might say that it would be an advantage to take opium production out of situations like this. You can see armed soldiers walking through an opium field in Afghanistan, and maybe move it into a situation like the one at the bottom where you have a bioreactor, and that would require taking the biosynthetic capabilities out of the poppy and putting them into something that you can grow in a big vat. Really, the driving force for doing this, though, is not so much controlling production, but if you want to make variations on an opioid, those are going to involve changing its molecular structure. And we're really surprisingly limited in how much chemistry we can do to a complex molecule like that after it's already produced. But if you can tweak the biosynthetic pathway, if you can change how the living system is producing it, you can change its structure and maybe do things like generate a pain reliever that's not as addictive. I think that would be a good thing. So to kind of address those goals, a team from Stanford University, among others, put together a bunch of genes from different organisms into yeast to cause it to make opioid precursors. And I just wanted to show all of the authors on this paper. There were 5 of them. Not a lot of papers in science in general, and not a lot of papers in synbio have authors that look like this, but it would be nice if there were more of them. So what this team at Stanford did, led by Christina Smolke, is they took genes from a whole set of different organisms. So this is the opium poppy here, but this is the California poppy. This is a bacterium. This is a rat. This is yeast itself, and this is another plant species. And they took all those different genes and put them together into yeast. And I'm minimizing the enormous amount of work that that takes. And I also don't want to alarm anybody by showing the pathway and the molecular structures here. But what I want to show you is that these different arrows represent different chemical reactions that take one structure into another. And those arrows all represent genes that come from these different species. So for example, the green genes are the ones that come from the opium poppy itself. But remember, we're actually doing all of this in a yeast host organism. So put together all these genes, do all this chemistry, and you can take sugar at the beginning through all of these molecular transformations and eventually end up with opioids and opioid precursors. And the real key of this is because you designed it from the ground up, because yeast is very easy to manipulate comparatively. If you want a different structure out at the end, maybe you can change some of the enzymes that produce it. So moving on to another very broad topic. So this is now programming microorganisms, but not to produce a molecule per se, but actually to do medicine of some kind. So there's one company, just to take one example, called Synlogic that actually has clinical candidates right now, where they are reprogramming normal gut microbiome bacteria to do things that can positively affect human health. So one example of that is, let's say you are a person who is not able to metabolize a certain amino acid properly. And so you build up that amino acid and that leads to toxicity, and you have to keep a very strict diet. Like one example of this is phenylketonuria. You have to avoid phenylalanine at all costs because you can't digest it. Well, one approach might be to try to change your own genes so that you can now digest phenylalanine. That's a pretty tall order. But it's much easier to add phenylalanine digesting genes to a bacterium that you can then take as a probiotic, essentially. And the bacteria can provide the metabolic functions that you don't have. That concept you can extend to all kinds of things where you are taking a microbe and essentially programming it like a computer or like a robot, a microscale robot, to do medicine within a person. And that might include destroying tumor cells or responding to markers of a disease and then giving some kind of report of them, or even producing drugs like the metabolic engineering, the cell factory example, directly in your gut at the times when they're needed. A related concept is using DNA circuits directly. So now not using the medium of bacteria, but just putting together parts of DNA, synthetic biology parts, to generate a circuit. So a circuit, just using the simple engineering definition, is something that puts together some inputs and gives you some kind of output. It has various ways of evaluating those inputs. So you might generate a circuit that evaluates certain molecules in a cell and says, okay, if you have this one and you don't have this one, et cetera, then you are almost certainly a cancer cell. If you can encode that kind of circuit in DNA and then put it into the cells in a patient's body, that circuit could be designed so that it leaves the normal cells alone. But when it's put into a cancer cell, it can cause that cancer cell to self-destruct harmlessly. This idea has actually been done. It works at least in a petri dish. It's a long way from working in the clinic, but this relies on the idea that we can generate these kinds of, small computing networks with biological components. Another thing I want to talk about is engineering our environment around us and trying to stop the spread of disease, not at the individual patient level, but at the population level. So many diseases around the world are spread by pests like mosquitoes. Malaria is a major example of that. And there are certain kind of bad actor mosquito strains. And Anopheles aegypti is a major one that spreads malaria throughout Africa and parts of the Americas. So using synthetic biology methods, if we can engineer the genes within a mosquito now, we can create properties of that mosquito that can lead us to either crash a whole mosquito population or potentially make those mosquitoes immune to carrying whatever disease it is that's of concern. So this is one example from a company called Oxitec. This is actually in field trials in all of the countries shown below and seems to be working in many ways. They have a synthetic biology circuit, again, putting together parts that come from nature, but in a new way, where their male mosquitoes always pass on a certain gene to their offspring. And that gene is harmless to males, but it kills the female offspring whenever they're born. And so the result of that is you engineer some of these mosquitoes, you release them out into the wild, and they mate with wild females, and then all of the children that they have are going to be male. Result of that is you have too many male mosquitoes, not enough female, and the population will go down. And on top of that, the male mosquitoes don't bite, only the females do. And so the immediate effect is to reduce the transmission of diseases from person to person. You can apply similar concepts to things like Lyme disease, where there's efforts to either make ticks immune to the Lyme disease pathogen, or to make mice allergic to tick bites so that the mice reject the ticks and break the life cycle that propagates Lyme. And there's actually trials like that that are taking place on the island of Nantucket. It's not exactly a synthetic biology method, but it uses many of the same tools. And if there's interest, and if I have time, there's some specifics I can talk about ways that you could sweep a genetic change through an entire wild population. A method like the one I'm showing you here is still a lot better than what we usually do to get rid of insect populations, which is to disseminate sterile insects out into the environment. We basically blast insects with radiation on huge scale. We need to cultivate many, many of them. We blast them with radiation so they become infertile, and then we release them out into the wild and hope that they will sort of act as decoy mates and not have progeny. But the problem is when you blast insects with radiation to make them sterile, you also make them sort of less fit, less interesting mating partners for the wild mosquitoes. So that kind of effort, although it works, if you scale it up enough, is really expensive and really ineffective. And this is a way to do it much more cheaply on a bigger scale. And last thing I want to talk about in terms of examples is actually a couple of cases from recent responses to the coronavirus. So some of you might be following the different vaccine trials that are in development. We're all very eager to get a vaccine in the clinic. And in order to support that, we are trying not just multiple candidates, but multiple candidates across totally different styles of vaccine. So those things might be using inactivated virus, so virus that has some of the antigens on it but isn't itself infectious. But many of the other ones are also using synthetic biology methods. So those include things like using a different virus, a non-harmful one, to put antigen molecules into the cells of a patient, or directly inserting DNA that codes for viral proteins into cells. So actually about half of the types, the whole categories of DNA of vaccines that are in the clinic right now use synthetic biology in one way or another. And then another example of this is in testing. So again, borrowing the really sophisticated, evolved technology that really, I mean, we can credit ourselves for discovering it, but basically we're just adapting it. It's already out there in nature. Is adapting a molecular detection system that's actually normally used by bacteria to detect a viral infection, and instead repurposing that so that we can detect SARS-CoV-2 in a patient sample and test for the presence of active disease. And so here, again, is maybe a more typical looking team from MIT that came up with this method. You're able to get a readout in something that looks a lot like a pregnancy test. So if you have two lines, you have disease. If you just have one, then you don't. And the way that that works is using molecular machinery, again, I would say evolved natural biotechnology from a CRISPR system to do this process very quickly, pretty much on the benchtop without any specialized equipment. All you need is a 60°C water bath and some tubes, and that's it. And the reason that that's possible is because nature has evolved these methods for amplifying and detecting biomolecules that really are far beyond what we can do synthetically. So there are ways to do the same kind of test to get the same kind of output, but you need a multi-$100,000 instrument, you need consumables that cost tens of dollars per sample, and you need skilled trained technicians and many hours of work time to get something that here is happening in 60 minutes. And again, that's because of the advanced technology that we have access to. And these are actually undergoing FDA approval process right now. So that's some examples of things that sort of more sophisticated grown-up companies are working on and research teams. I also want to mention the highly enthusiastic and maybe somewhat less skilled student synthetic biology community. So there's this fantastic organization called the BioBricks Foundation that sponsors the International Genetically Engineered Machine Competition every year. And this includes many thousands of students from countries all across the world, and that number of countries is expanding every year. And those teams of students use what they know about synthetic biology to try to address real world problems, sometimes ones that are very specific to the communities that they come from. What I love about those teams is that they don't yet have the pessimism that professional researchers do, so they don't know that they shouldn't even try to do something. So oftentimes they're thinking way beyond what we're capable of really achieving. But you know, the field is advancing and it's still very new. So oftentimes these projects are incredibly creative and potentially really impactful. And that can be seen in part by how much money those teams have been able to raise. There's a huge alumni network. So I've been involved in iGEM, not as a student, but as a judge. And many of the now people who are becoming principal investigators or graduate students or industry people have come through iGEM in one way or another. And that's also manifested in over 20 of the projects over time have become startups, including some tech unicorns like Benchling, which is the platform that I use to do my DNA design. That came out of iGEM many years ago, and now it is a full-fledged platform. So that's many of the optimistic things about synthetic biology. I do want to mention that as with GMOs, there are many people who are concerned, legitimately, despite what this picture might make you think, with the risks of synthetic biology. So synbio is creating new things from previously naturally existing parts, and sometimes it's adding to those parts, making things that haven't existed in nature before. And certainly, in the kind of GMO examples that I gave you before, there is kind of a difference between just disabling one gene in an organism and putting genes together from all different areas of life. And so there's both a legitimate and reasonable concern about the risks of synbio, especially because it is a self-propagating technology, you know, because life can reproduce itself. And at the same time, there's a lot of alarmism, and the balance between those two things is going to, I think, affect how good the outcomes are for investment in synbio and its application to actually do good in the world as opposed to less good things. So I've told you a bit about what's happening outside of Williams College. I want to briefly mention the work that's going on in my lab and how students are trying to help out with advancing the synbio field. So we are aiming to develop a new platform for synthetic biology. The organism has a Latin name. Peter actually did a great job with the strictly Latin pronunciation, but everybody who works on this bug says it a different way. So I usually say VNAT, which is snappy and short. So we're trying to learn more about this bug and start to switch over to using it for a lot of the things that people currently use E. coli for. So why is that? So E. coli, you're seeing it here, growing and dividing in a little microfluidic chamber. This is the bacterium that pretty much everybody uses for whatever they want to do. It's not necessarily the best at every kind of application. Like if you want to do metabolic engineering, you want it to crank out some sophisticated molecule, it may not be the perfect choice, but it's very easy to engineer. And so labs use it to put their DNA together. They use it to make proteins like insulin. This is the same bug that was used to make human insulin and still is, and to make synthetic biology devices. Why is that? A few reasons. So first off, it grows pretty fast. So the video you're seeing here is sped up, but the doubling time, each of those cell divisions is happening within maybe 25 minutes at the fastest. And that's pretty fast because you can see it grows and divides exponentially. The other thing is it's pretty easy to genetically modify. So we can put DNA into it, take DNA out of it, mess with its genome. And really, the biggest thing is people converged on this organism really early on in the field, and people have studied it so much, starting as early as 1920. And in modern times, there are these fantastic databases and resources that will tell you all about what's going on with E. coli and its DNA, like ecopsych.org, which is this huge database of literature curation and understanding of how E. coli works. So that's E. coli. Here at the bottom is VNAT. And you can see right away, there is a difference in how fast VNAT grows compared to E. coli. These videos were actually taken under exactly the same conditions. These channels are right next to each other. They're both in their optimal growth medium and temperatures. And so VNAT is able to grow and double in times as short as 10 minutes, maybe 15 at the longer side. And that speeds up everything. So your workflows that involve putting DNA together can be sped up by that. Many of the methods that you use to do synbio, the cycle time to go around that design build test cycle is limited by just getting your bacteria to grow. It's similar enough to E. coli that many of the parts and devices that we already understand will carry over, but it's also kind of different. And so it's kind of unclear how much adoption it's going to get in the synthetic biology community. There's a certain amount of skepticism. It does grow really, really fast. And it has some great methods where you can easily change the DNA on its genome because of, again, things that it's actually just evolved to do. We didn't invent them, but that function is there in nature. Problem is, it was first reported a long time ago, but nobody really studied it since about 2015. So you compare that to the history and the accumulation of knowledge around E. coli, which, by the way, we still don't understand as well as I would like, and there's a certain gap there. Now, we're learning about VNAT and about all kinds of bacteria and other organisms much faster than we did back in the 1920s or 40s or 80s, but there's still a lot to do. And so what my lab is working on, broadly speaking, is learning more about VNAT, figuring out methods to work with it and engineer it, figuring out how all the parts and devices that we already have are going to perform in that setting. And then we really want to take advantage of its special properties. So its ability to evolve fast so that we can change the kinds of things that it does, its ability to be a microbial cell factory to pump out different kinds of molecules, because if you grow really fast, you have to be able to process nutrients really fast and make things really fast. And of course, its growth rate. So again, without getting into the details of how that's going to go, our main mission is to try to get VNAT to replace E. coli as the standard lab organism. Who knows how that's going to go, how long it's going to take, but I am excited about it. So I hope that all throughout I have presented you with many challenges and opportunities for synbio, but I just want to highlight a couple of them here at the end. I already mentioned just now that our lack of knowledge is a big deal. And here's a paper from 10 years ago now that identified 5 hard truths for synthetic biology. Whether living systems really can be engineered, whether they're engineerable. And I summarize those things as follows. So many of the parts are undefined. So when I say, here's a DNA sequence, here's what I think it does, that's not a trivial thing to do. That's where you actually need a biochemist, a molecular biologist to get in there and understand it. There may still be a few things left to learn about biology. And then the design, build, test cycle is great, but we're not necessarily very good at designing, building, and testing. And so we have to go around that cycle many, many times. The more complex the system that we try to generate, the more likely it is to fail. And so all of the work that I've shown you so far, the simplest things took a lot of effort, and the more complex ones took many, many person hours over years to make happen. And then really, nature doesn't want to be engineered. It doesn't want anything. So our goal of making things modular and having abstraction and so forth doesn't necessarily fit with how things evolve. So we have to kind of push those naturally occurring things into that mold if we want that to happen. And then this one is starting to get under control, but biology has a lot of randomness in it, and it's not quite the same as transistors on a chip. And so we need a true molecular level understanding of how it works. And this is why, you know, as a chemist, I think it's valuable for people like me to be doing synbio, to get a handle on those events and how we can make our designs and our systems robust towards them. So I want to thank all of you for listening. I want to take some questions, which I guess I will select in somewhat random order, but starting from the top. I want to thank the students who have worked with me. I don't have a picture of the summer students who just started with me a couple weeks ago working remotely, but here's Dylan Milson, who just did a thesis this year, Vanessa Ayan, and Madeline Ohl. And I'll leave you also with this cartoon for a moment, which is suggesting that there may be a few difficulties in engineering biology. And I'm going to take a look at your questions. Please keep sending those in. So here's one from Daniel Gastel. Sorry if I mispronounce everybody's names. So Daniel asks, are DNA sequence database labels crowdsourced? And if so, what protocols are followed in updating DNA sequence databases to ensure the updates are correct? So Daniel has identified an enormous problem with this field. And I actually will take the chance to show you a couple slides about that. So the way that we used to answer questions like that is people wrote papers about it. So even as relatively recently as 1992, if you wanted to learn about some biomolecule, like this one has a very simple function, it binds to a small molecule and that causes it to stick to DNA and turn on the expression of some gene. So that's all it does. But how do you learn about that? Well, you look at papers from 1992 and they have table 2 and they have a certain strain and a certain plasmid and then another plasmid. And then there's a table of values and how did they wire this up and what were the conditions and so forth. So you can picture that that is a non-trivial challenge to get through. The goal for synbio is to crowdsource that kind of process. So bringing these kinds of results into the modern era, getting them in formats that are easier to parse and understand, and also to update some of that data with the understanding that we have now. And so this is the dream, is you have some kind of data sheet and say, here's your biomolecule. It does this. It's a receiver for polymerases per second. Here's its response curve under standard conditions. You add a certain amount of the small molecule here and the response goes up. Here's how it performs over time. Here's other things you can wire it up to make a circuit. And so forth and so on, all in a standard format. These things just don't exist yet. So this was literally presented, again, I was mentioning the BioBricks Foundation. They have a registry of standard biological parts where they want people to put things like this in. That's the registry that iGEM teams deposit all of their results and information to. And I think that's fantastic, but it doesn't look like this necessarily. It looks like a dump of unstandardized data, often run by people who don't quite know what they're doing yet. And so getting this information is not easy. And I hope that it gets easier in the future. So the kind of annotation and informatics challenge is real. This is just an example of a page from the registry where literally what I showed you on the earlier slide is marked out. And this is something that anybody can edit as a wiki and contribute to. And hopefully we're getting better over time. So I have a couple questions here that maybe I can address together. One from Robert Hallman, one from Stephen Kronheim. Those are surrounding the question of if you engineer a wild population, if you make a change to that community, how do you know what the downstream effects are going to be? That also is a really hard problem. And I didn't mention it up front, but again, there are ways to introduce changes into a population that are even more drastic than what I was describing. So here's an example of that where if you have certain genetically engineered mosquitoes, the ones that I showed you just cause the population to crash. But there's methods using CRISPR where you have an engineered mosquito and a wild one, and all their progeny will live, and all of the progeny will get the engineered genes. So this is something called a gene drive, and the promise of it is you can use it to change a population out in the wild, not in the lab. There are huge risks with doing that. And in fact, the person who sort of first publicized gene drives, Kevin Esvelt, then has completely changed his mind about whether they're a good idea to use and has been publishing things publicizing the risks. So the trials that are in place right now, there's a certain bias towards doing these things on islands. So for example, the one that's in Florida is in the Keys. These are the Cayman Islands is another test site, because you want to limit the release and see what happens. I think this is a real area of controversy, and my personal perspective is you have to consider the base case. So how many people is it acceptable to have dying of malaria versus whatever downstream effects we might have? We want to have pretty good models, but especially with things like climate change, we're already intervening in a pretty big way in what's happening out in the world. And so it might be worth taking some risks. The tools that are developed to assess those, that's where synthetic biology needs close and tight involvement with other disciplines, because synthetic biologists don't tend to be ecologists as well, or at least many of the people doing this work are not. And so places like Oxitec are trying to engage with stakeholders, trying to engage with people who understand wild mosquito populations, for example, before they just go releasing these things into the world. So here's a question from Andrew Waldo, asking, the European Union and the US have taken very different approaches to the regulation of GMOs, and asking what, in my opinion, are the appropriate ways to regulate the unintended consequences. So the European Union, by and large, has decided that it's the product of genetic modification, not the process, that makes something a GMO. So in that situation, the ruby red grapefruit, which has just been irradiated, no longer has any foreign DNA in it, and so it's not a GMO. And neither would be an organism that you make using CRISPR to edit the genome of something precisely. If you don't stick any new DNA in, you just cut DNA out or introduce some mutations, then you don't have a GMO there. My attitude, and the US has taken the opposite perspective, which is if you use CRISPR, then it's a GMO no matter what. Even if you made a very precise, well-characterized change that precisely mimics something that already exists in nature. I think we really have to look at the safety characterization and the expected effects. I don't think you can just label something as a GMO or not based on simple criteria, because sometimes a relatively small change can cause a big risk. For example, if you're modifying something that's pathogenic, then a one base change in a genome could cause a strain of the flu to cause a pandemic instead of not. And that process means strong scientific involvement in the regulatory regime, not just a hard and fast rule. Sorry, I'm just paging through the questions here. Lots of great questions. And this is the problem with getting to choose which ones I want to pick. So kind of a question from Robert Graham. How do you get bacteria to receive and replicate a new DNA sequence? This is a good question getting into the logistics of synthetic biology. It's a big reason why I work in bacteria and why many people do. That capability of putting new DNA into something and having that DNA be replicated is sort of core to everything that we do because we work at a genetic level. And oftentimes the methods for doing that are pretty unsophisticated. So if you want to get a bacterium to put some new DNA into its genome or into its cytoplasm, you often will either deliver it a very strong electric shock, and then the bacteria that don't die sometimes have DNA jammed into their cell membranes because DNA is a charged molecule, so it'll move with that current. Or you can put them in a chemical bath that will make their membranes so porous and full of holes that DNA can just float in sometimes. And then the very few of them that survive that process will have the DNA inside. So those things actually work great. I don't mean to minimize them, but it does place some limits on what you can do. One of the reasons I'm excited about VNet in particular is this organism has sort of a rare characteristic, not that rare, but rare in model organisms, where it has evolved to actually pull DNA in from the environment. So these are some fluorescence microscopy images of a Vibrio cell sticking out a pilus, just a chain, a tube essentially coming out of the cell wall and latching onto this DNA molecule labeled in red. It reels it in, and then not shown here is that it will actually stick it into its genome, provided that DNA molecule has sequences on it that match up with the genetic sequence. And so that process is really gentle because it's there in nature already, and again, more sophisticated than what we can do by just treating things with chemicals or zapping them. And so there's some implications for evolving VNAT quickly that could enable. A couple of questions coming in from Thomas Vitali, and there was another one from Daniel Gastel about what kinds of genetic diseases might be targets and how that works. So CRISPR is causing a lot of excitement right now because it efficiently lets us incorporate DNA into a genome. You still need to deliver all the CRISPR machinery into a cell if you want to change that cell's genome, but we now have the capability of making really precise changes. So changing one site in a genome and not any other site in a whole range of different organisms. The real challenge there is not so much about what things we could correct or target using CRISPR, because if anything, the possibilities are pretty limitless. Any disease that has a primarily genetic cause, like a metabolic defect, like many inherited diseases, of course, those are things that you can potentially correct with CRISPR. The real challenge is not so much finding the targets, but in getting the machinery into cells efficiently and making sure that you get the edits that you want and not any edits that you don't. So CRISPR is very, very precise, but it's not necessarily precise enough when you're thinking about a human patient, because some changes to the genome might lead to giving you a tumor, giving you cancer. And so, or if you're doing editing that might be passed down to future generations, that's especially of a concern. You don't want to edit germline cells. So the delivery challenge is really huge. And there's also many people who are investigating ways to make CRISPR even more precise than it is now so that you can do those therapeutic applications. CRISPR at the moment is really a fantastic research tool. So it lets people do engineering on human cells in a petri dish to answer questions, like, for example, to test things about COVID-19, SARS-CoV-2 susceptibility. People have used CRISPR to really rapidly screen through what the different kinds of genes are that might increase or decrease your risk. But in terms of its clinical applications, it's definitely going to be further behind, and the delivery challenge is the reason why. All right, so I'm going to take just one last question before we wrap up from Ginta Remiekis, sorry. This is about what safeguards are in place to monitor for ethical concerns and challenges. And this is my plea to all of you. So when you go out in the world, learn a bit more about synthetic biology, about what things are happening. Make sure that the people that you elect into public office seem like they know what they're talking about, because there's a lot of poorly informed opinions about these things on both sides of the issue. And I would much rather have experts who are grounded in the science, but also responsive to a wide range of constituencies. So not just using synthetic biology technology to advance people who already have a lot, but to make the results a bit more equitable, and who are going to make science-based decisions about them. And like I said, it's especially concerning when, for example, in the US, on the political right and the political left, there are people who are very worried or who are very boosting synthetic biology. So I'm hoping that we can get scientific expertise in there. And that starts with getting an informed citizenship and with people caring about this and wanting to learn more. And I'm happy to talk to anybody who's interested about that. My email's on the Williams College Chemistry Department page, so drop me a line and I'm happy to talk more. But I think for right now, we are out of time. So I'm going to turn it over to Peter to wrap up. We might have lost Peter.
Hello, Peter. Okay, well, thank you so much, Professor Thuronyi, for everything, for answering all those questions. I looked at those questions. They were difficult questions for me to understand, so I appreciate you breaking everything down for us. Peter and Alicia Pond have hosted the Pond Lecture for 25 years. I believe this will be our 25th year. This would have been. So next year, we will be celebrating 25 years. And we look forward to all of you all joining us. Thank you so much for joining us today. Please log on to the alumni.williams site to find out what events are coming up next. And we look forward to seeing you at the remaining lectures for the summer faculty lecture series. Thank you all so much. Thank you, Professor Thuronyi. Thank you very much for your time.
Thank you, Ben. Excellent job. Wonderfully done.